Page 49, we have got the diagram shows some tissues in a leaf. So that's the top of the leaf, bottom of the leaf. And we can see stomata. We've got the upper epidermis, the palisade mesophyll layer, spongy mesophyll layer, and then lower epidermis with the stomata on. Name 2 organelles in a palisade mesophyll cell that could be seen with an electron microscope but not with a light. So with a light you can see kind of nucleus, you can see chloroplasts. You can't really see much else. You might be able to see mitochondria. I don't know whether I've ever seen this, but let's pick some really small stuff like endoplasmic reticulum or ribosomes. So let's have rough endoplasmic reticulum, and then let's have Golgi body. Part B, the palisade mesophyll is adapted for photosynthesis. Okay, 2 ways in which the cells are adapted for maximum light absorption. Well, it doesn't show you here, but the chloroplasts will be in there, so we can say contains chloroplasts, and these chloroplasts are kind of near the edge of the cell, near the edge of the cell. Also, we can say these are arranged, almost at the top of the leaf. We could say the chloroplasts can move in the cell. So, cells arranged near the top of the leaf. We could say, if this was another point, chloroplasts can move to the light in the cell. Efficient uptake of carbon dioxide. Well, here we're trying to use this diagram really. We can see there's some air, some air spaces, not as much as the spongy mesophyll, but some air spaces around for diffusion of gases. So some air spaces around the cells for diffusion of CO2. And we could possibly suggest that they've got kind of a large surface area to volume ratio, larger than the spongy mesophyll. Or we could say they are, there's air spaces from the stomata. So let's have a large surface area to volume ratio. Say near to the stomatal space, or there's air, the passage of air from there to there. So really trying to use the diagram to come up with sensible suggestions.